Okay, so picture this. A new virus emerges, and the clock starts ticking. The whole world is in a race to find a cure. But right at the heart of that race is this huge problem. You have to find one single, perfect molecule that can stop this new threat. The question is, where do you even start looking?
Trying to find that one specific molecule is like looking for a single, unique grain of sand on every beach on the entire planet. Because when we talk about fighting a disease, the number of potential weapons your body has in its arsenal is just, well, it's staggering.
Yeah, that number right there, 10 quadrillion. That's the theoretical number of unique antibodies your own immune system can create. It's this almost unimaginably huge library of biological tools. And here's the crazy part. Somewhere in that library is the key to stopping the next pandemic. The problem has always been, how do you find it?
So how do you even wrap your head around a number that big? Probably the best way is with a simple analogy. Just think of your immune system like a giant keychain with trillions and trillions of keys. And when a new virus shows up, it's like a brand new lock. The challenge is finding the one single key on that massive chain that fits perfectly.
Our bodies are amazing machines, for sure. But for scientists, trying to sift through this massive biological library has always been a huge bottleneck. It's technically really demanding, it's incredibly expensive, and it can take months, sometimes even years, to find the right antibody.
All right, quick refresher just so we're all on the same page here. An antibody is basically this little Y-shaped protein that your body makes to find and tag invaders. Each one is custom shaped to fit a very specific target, just like a key is cut to fit only one single lock.
But what if you could teach a detective, a super smart detective, to instantly spot the right key just by looking at it? Well, that's exactly what a team of scientists decided to do by training a whole new kind of AI.
They call it ASPRED, which is short for Antigen Specificity Predictor. And its entire job is to be a master hunter, an expert detective that can scan millions of these antibody keys and predict which one is going to fit the viral lock.
So how in the world does this thing work? Well, think about the autocomplete on your phone, right? You type a few words and it predicts the next one. A protein language model, or PLM, does something really similar, but for biology. It analyzes hundreds of millions of protein sequences to learn the language of proteins, the fundamental rules for how they're built and what they do.
So ASPRED is one of these PLMs, but it's been fine-tuned for one super specific job. It reads the amino acid sequence of an antibody, its basic code, and based on all the patterns it's learned, it predicts whether that antibody is likely to bind to a specific virus, like SARS-CoV-2.
Okay, so that sounds awesome in theory, right? But a prediction is just a prediction until you can prove it. So, the researchers put ASPRED through a whole series of tough challenges to see if it could really walk the walk.
And the results? Whoa, this was a huge leap forward. When they tested it on SARS-CoV-2 antibody sequences, ASPRED hit over 80% accuracy. The best model before this, it was stuck at about 55%. That's not just a little bit better. That is a total game changer in terms of predictive power.
But they didn't stop there. To really kick the tires, they compared ASPRED's digital predictions against a top-tier lab method called BEAM, which can physically test thousands of molecules at once. And get this, the list of antibodies the AI flagged had a huge, statistically significant overlap with the ones found in the real-world lab experiment. This thing was finding the real deal.
All right, next up, they threw it into a real, messy biological system. They looked at the entire immune response from mice that had been immunized against SARS-CoV-2. And out of more than 300 unique antibody sequences, ASPRED cut right through the noise and flagged just 30 as the most promising candidates.
Okay, this is where it gets really cool. Could they actually take these digital predictions from a computer and turn them into real working proteins in a lab?
So the process was pretty straightforward, really. 3 key steps. First, let the AI, ASPRED, do its thing and predict the best candidates. Second, scientists would actually create or synthesize those proteins in the lab. And finally, the ultimate test, would they actually bind to the virus? So, to run the experiment, the team just randomly picked 10 of the candidates that ASPRED had pointed out. They created them as nanobodies, which you can think of as smaller, simpler versions of antibodies that are just way easier to produce and work with in a lab. And the results? Absolutely stunning. Of the 10 candidates they picked at random, 5 of them, that's a full 50%, showed strong, robust binding to the SARS-CoV-2 spike protein.
You have to understand, a 50% hit rate from a computer just predicting, that is phenomenal in the world of drug discovery. It's an incredible success.
And within that group of 5, one of them was a true superstar. We'll call it AB157. And this number, 20.7 nanomolar, that's its secret sauce. But what does that actually mean?
Well, it's a measure of something called binding affinity. It basically tells you how tightly the key fits into the lock. And here's the most important part you need to know. A lower number is better. A lower number means a stronger bond. And at 20.7 nanomolar, we're talking about a very, very strong bond, absolutely in the range you'd want for a potential new drug.
So, let's pull back for a second. What does a tool like ASPRED really mean for the future? Because this is way bigger than just one virus. This is about changing how we find new medicines, period.
This whole approach basically changes the game in 4 huge ways. First, speed. We're talking about crushing a timeline that takes months or even years down to just days. Second, scale. It can analyze millions of antibody sequences on a computer. Third, generality. It's not just for one virus, it's already shown it works well for influenza and HIV too. And finally, efficiency. It slashes the need for all that expensive lab work by pointing scientists directly to the best shots on goal.
You know, the researchers themselves put it best. Their big takeaway is that everything you need to know about what an antibody will attack is encoded right there in its sequence. And once you realize that, it just unlocks this incredibly powerful and scalable new way to discover life-saving medicines.
And all of this leaves us with a pretty mind-blowing final thought. We just saw an AI that can hunt for cures that are already hiding inside our own biology. So the next logical question is, if AI can find these molecules, could it one day learn to design brand new ones from scratch, perfectly tailored to defeat any disease it comes across?